Let us use the reactions button there where it says reacts and put our hands together as we invite Sue, our instructor, to take us on module 6. Thank you, Alvin, Joyce, and Edith. I'm also delighted to welcome Sue.
Hey, James.
Hello.
Thank you for that great introduction. Hello, everybody. Good to see you all again here today. And to any of the new students, you've come very late to the party, which means that there's a lot of stuff that you haven't quite gotten yet, okay? Today, I'm going to be taking you through, you can't see it here, actually, I'm going to take my blurred screen off so that you can see my background. So this is my laboratory, and you can see my microscopes in here. There's 1, 2, 3 microscopes there. Today, I'm going to take you on a compost safari, a microbe safari with my microscope. But before we get there, we need to know what are we going to be looking for. So we learned about the importance of the fungal to bacterial ratio. We learned how critical it is in terms of, the different succession of your plants, whether you want to be, growing brassicas as an example, where the fungal to bacterial ratio is not that important. But if you want to be growing grains, grasses, trees, shrubs, fruits, etc., avocados, you need to really have that right fungal to bacterial ratio in your compost, or get them the best way you can. So today we're going to do two things. We're going to go through a small presentation that's going to show you what fungi looks like under the microscope. And then at the end of the lesson, we're going to go to a small safari here. We're also going to do the introductory part of compost making. So lesson 1 and 2, they're both 20 minutes long. But basically what it's going to do is teach you about the equipment and what you need to make compost, and then how to make the compost. So I hope you've got pen and paper. You're going to take notes because it's quite important. In your homework and in Canvas, you'll see that there's also part 2 of the James White webinar that we were watching yesterday. Please go ahead, watch the rest of that webinar, because we don't have time to go through it in the course now, because I want to do this with you. But it is extremely important and very, very informative. So I'm going to share screen with you now, and I'm going to take you through the fungi part of this course.
Yeah, the screen side. Yeah, they are changing.
Great. So remember this screen that I've been showing you all the time, and I've told you you'll see it time and time and time again, right? And this is the soil food web. We, in the second trophic level there, the bacteria and the fungi, this is what we're focusing on today, hugely important. Some nematodes in that space as well. Nematodes can be really tiny, right the way up to earthworms. Earthworms also considered in that range of nematodes. But we'll start by reminding ourselves the soil food web, it defines for us who eats who is dependent on who, and how collectively all the creatures in the soil stabilize the soil so that we can grow healthy plants. Soil food web is divided into trophic levels, with the first being the photosynthesizers, the second trophic level, the decomposing mutualists, pathogens, parasites, and root feeders. Fungi are part of this group. Now, in her book, For the Love of Soils, Nicole Masters describes fungi as being the organisms holding the fabric of soil life together. Okay? And that what sets fungi apart from other kingdoms of life is chitin. Okay? Chitin is what makes up their body, which is a long-chain carbohydrate found in their cell walls. And that is basically made up of a lot of carbon. Okay? The fungi are underground. The fungi and the other microorganisms act like our underground livestock, but fungi in particular like our underground piping and moving systems. Okay, an individual hyphae, that is one tiny strand. An individual hyphae, as an individual hyphae, they're invisible to the naked eye. They're only 2 to 10 microns wide. That's very tiny. But as they adhere and bundle together, they become the visible mycelia, the structures that you can see in this picture, the white mycelia on decaying wood. They're basically going in and dissolving and recycling every single part of what was in that wood, and in the process building their own bodies and moving other nutrients within and on their bodies. Nicole Masters gives us a good visual reference. She is an independent agroecological systems thinker and educator. And she references fungi by further describing the soil fungi as being like the cement and stones that build our homes. They're a vital part of the foundation of the soil structure. They're also the neural network of the soil, sending information across thousands of acres, communicating insect attack, instigating plant defenses, and sharing resources. Estimates point to at least 70,000 species of soil fungi across the world. These include yeasts, molds, mushrooms. However, the figures just estimate. Many, many more are unknown. Okay. Soil fungi groups can be categorized as having 3 major functions in the soils: mutualists, saprophytes, and pathogens. The first group, mutualistic fungi, offer an array of benefits to plants and animals. These are examples, and there are examples of animals that choose to partner with and even farm fungi, as you can see here with the termites. These include leafcutter ants, stingless bees, beetles, and termites. The termites cultivate the fungi in a fungus garden, comprising a few hundred fungus combs, structures built from chewed up grass and wood and inoculated with fungal spores. So where we do the same thing to produce mushrooms, ants are doing it in the ground. Each year, these fungi produce a crop of large mushrooms. In some areas, these mushrooms are known locally as omajoa, which are highly prized as a delicacy. The ants do it because they feed their young with parts of those fungi. So they farm them for food in the ground. It's craziness, eh? Then we've got the lichen. We all see these on our trees outside. Lichen and algae require this mutual relationship with many species of fungi for their survival too. Next time you see lichen in your environment, take a very close look to see if you can see their fungal support system. Yeah. Lichen are a complete life form that is a symbiotic partnership of 2 separate organisms, a fungus and an algae. The dominant partner is the fungus, which gives the lichen the majority of its characteristics, from its shape to its fruiting bodies. That's pretty cool. Mycorrhizal fungi, meaning fungal, and rhizal, meaning roots, meaning fungal and rhizal, meaning roots, are mutualists which live on and in plant roots, creating a symbiotic relationship between the plant, fungus, and the bacterial partners. The mycorrhizae reduce plant stress. They offer root protection and water and nutrient uptake in exchange for carbohydrates, carbon, energy, and fatty acids. Mycorrhizal fungi are those that go into, most of them, into the actual cells. They're endophytic. Dr. James White spoke to us in the picture of the blue fungi, the endophyte that he told you was a fungal endophyte very early on in his presentation. We can consider mycorrhizal fungi as fungal endophytes. Their hyphae are much finer and can travel much further than plant roots, increasing the surface area of the plant for water and nutrient uptake by up to about 40 times. They're a significant component of healthy soil ecosystems, making up as much as 1/3 of the microbial biomass in healthy soils. Over 90% of plant families of 250,000 plant species have this mycorrhizal relationship, supporting healthy, resilient plant growth. Then you've got the saprophytic fungi, okay? The saprophytic fungi are the largest group of macrofungi responsible for breaking down and recycling dead plant and animal material. These are the fruit bodies you see on dead trees, leaf litter, animal bones, and even feces. Saprophytic fungi release enzymes to break down and digest the lignin, cellulose, or chitin in this material into simple, soluble compounds that can be absorbed by them and by plants as nutrients. In so doing, they play a vital role in reducing the accumulation of dead organic material and in recycling essential nutrients, particularly carbon and nitrogen. Without these digestive activities, forests would disappear under a mountain of logs and leaves. They basically keep crunching stuff down and bringing it back into its basic element, so it's recycled back into systems again. As you know, forests and other ecosystems are growing from the plant biomass above that is photosynthesizing, and it's not the soil surface that is rising up to cover the plants. That wouldn't be a very good evolutionary strategy. Pathogenic fungi are fungi that cause disease in humans or other organisms. About 300 fungi are known to be pathogenic to humans. Markedly more fungi are known to be pathogenic to plant life than those of the animal kingdom. The study of fungi pathogenic to humans is called medical mycology. We've all heard of athlete's foot, candidas, and ringworm, and in plants, we're all familiar with fusariums, mildews, rusts, and botrytis. Mushrooms are the fruiting bodies of some fungi. Let's look at how fungi reproduce. Most fungi reproduce asexually through asexual reproduction. This means they do not need a female and male connection, but rather the young get all of their genes from a single parent. Although fragmentation, fission, and budding are methods of asexual reproduction in a number of fungi, the majority reproduce asexually by the formation of spores. Here we can see a variety of fungal spores as seen under a microscope. Okay, we're going to be going under a microscope. This is what we're going to be looking for. As you can see, they come in a variety of shapes and colors. These are pictures of spores that were taken here in my lab. The first is of wild mushrooms that had sprouted after the rains. The second two were seen in a compost and a vermiculture sample. Their large size and dark color indicates that they are from good soil fungi and perhaps even mycorrhizal fungi. Fungi are everywhere. Here we can see them in a packet of tomato sauce. We looked at the green mold on the top of the fungi under the microscope and saw that it was in fact green spore. As we can see in some of the pictures above, the pathogenic fungi are not desirable. When we look at these under the microscope, we can see some very, very obvious traits. Some of this includes color, diameter, cross walls. These are quick indicators of good or pathogenic fungi. So when we're looking under the microscope, this is what we're looking for. Most good fungi are colored from reds to dark browns. They also have very clear wall separations. These are called septa, which are usually spaced at equal distances. Good fungal hyphae are also quite wide compared to pathogenic ones. Pathogenic ones tend to be colorless, grouped in crazy, messy clumps. They have irregular spaced septa, and they're quite narrow, and they have very fast growing reproductive parts on them. You can see in the picture on the right what a mass of non-desirable fungi look like under the microscope. The reason they are like this also is that they grow extremely quickly because their time is short. They don't survive for very long because we live in an aerobic world, and the minute things do switch through competition, inhibition, and consumption, the environment around them will flux and change very quickly towards aerobic, and they won't survive. So they do grow very quickly, and they also disappear very quickly if we give them the right conditions. When we look through a sample, we aim to identify which fungi are present. If you look closely, you can see a tiny one in the middle of this picture. We'd need to increase the magnification to 400x to identify it well. Here are some good fungi, nice brown color, clear and evenly distant septa. Some of them are wide. And here are some of the pathogenic fungi. We know now that fungi are great decomposers. They therefore hold nutrients locked up in their structure and are also predated on by other organisms in the soil food web. And these organisms cycle these nutrients back into the soil in plant and other microbe available forms through what we call the poop loop. So you can see there at the beginning, the sun, the photosynthesizing, living plants, dead organic matter, the decomposes, that first, second actually, trophic level, going on to all those others, the microscopic soil predators, protozoa, amoeba, flagellates. As they break things down, they release nitrogen and it goes back into the poop loop. Yeah, back around in this poop loop. Finally, let's keep in mind that soil health is dependent on soil microbial diversity and maturity. With this in mind, we're going to now come out of this presentation and we're going to go onto the microscope. We'll do that before we look at compost. What I have in my lab, and if you've got any questions, please just throw them into the chat on the side there. I have a pot of compost. This is what it looks like. This is a 6 to 7 month old compost. I made it, or maybe not, maybe it's about a 5. I've got the records of it somewhere. It's really important we all keep our records. I think it's about 5 months old. It looks a little bit like soil, but you can still see some woody bits in there. We add more woody chips when we're making compost so that there's porosity so that the compost can actually breathe. I've put 1 cubic centimeter of compost into this test tube with 4 cubic centimeters of water. So it's up at 5. And I'm going to take 1 drop of it out. I'm going to put it on a slide. Just going to get my in my slide. I'm going to put 1 drop on the slide, and I'm going to put a slide cover on it, and then we're going to look at it, and I've got my microscope connected to my computer. I'm going to switch to that software. Just before that, show you what I'm doing here. So I've got 1 drop that I'm going to put on there, just 1 drop in the middle of the slide. Down to my leveling it. There we go. That's 1 drop. No more, no less. I'm going to clean out my pipette. We keep these things clean. I'm dropping the cover slide now onto that 1 drop. And now I'm going to put it onto the microscope. I say goodbye to you here. And we're going to go, I'm going to now share. I'm going to go directly to share with you what's under the scope. And I'll keep talking to you and telling you what I'm seeing. Let me bring that into let's move it into right away. James, can you see this well?
Yes, I can see so.
Perfect. So it doesn't look so wonderful, this kind of view. You're probably wondering, what is all that? What is that dark stuff? That's what it looks like. So a whole bunch of decomposed things. I'm trying to make it a little bit more focused for you. Now, between the two slides, between the slide and the slide cover, there is about 9 micromillimeters. Every single bacteria is about 1 micro. So there's basically enough space to have 9 bacteria in a line. When we look at some of these microbes, they can move in and out of actually, it's almost like a swimming pool inside there, as much as it is so tiny for us to imagine and see. This microscope is a 10 on the eyepiece, and this is a 4 down here, so that's at 40x, we say. I'm now going to move it to a 10 and a 10. Does anybody want to tell me in the chat what overall magnification we're looking at? 10 and 10? We got any answers there? I can't see.
Not yet. If you can repeat the question, or what do you want them to write?
So the asking here is, if I have 10x magnified by 10 in my eyepieces and 10 in my objective lens, what is the overall magnification that we're looking at now?
Yes, team, we can put our answer in the chat area.
Have you got any answers there?
Yes, we have the first answer.
And what have we got?
100.
Who was that? 100%. Now,
Vivian.
we're now going to actually go even deeper. We're going to go into 10 on the top, 40 down, which gives us how much?
Great.
What have we got? Any answers? What are we looking at now?
Edith. Got an answer from Edith. Anyone else?
What has Edith said?
200.
No, we're looking at 40
Yep.
on the, we're looking at 40 and 10.
Yes. 400x, Joyce, Chai, and Edith.
Perfect. Now in there, I see something moving, but in there, if I try to zoom in, you can see that piece of fungi there. Let me just show it to you. So this here, where have I got my tools? This here is a piece of fungi. You can see when I zoom in and out, it has a scepter over there. It has another, I think there's a scepter over here. I'm going to just take that away. And I'm going to zoom in and out of it slowly. Scepter up at the top. Right down. There's a bunch of other stuff in there. It's not very clean. This I'm going to take us to the edge of this particular slide, where maybe we're going to see, sorry, over here. Sorry, that went a little bit fast. We will see things a little bit clearer. Look, there's another piece of fungi in there. Oh, and there's a whole other piece as well. Let me bring those into the picture. More. Oops, let's get more light. Oops, sorry. What is happening here? So there's a very big piece of fungi down the side there. right here. But there's also some more in here when I zoom in out. See that little bit in there?
Yeah.
Now, all of these little dots that you're seeing, these little dots that are moving, these are all bacteria. So that's how small they are. Okay. Now I want to just do something here. Okay. I want to do that. I want to just try and take away these 2 points I just put in there. And let's just see something else. What else might we see in here? A very nice piece of fungi just coming into the middle there. Can you see that?
Yeah.
Very nice right there, branched fungi. See, I'm not seeing any of that white, messy stuff. I'm seeing nice brown fungi. When I go down a little bit here, I'm seeing some more up in that corner across there. And then there are some other interesting things also. Oops, let me move this up this way. So right here in the middle of the picture, right here is an amoeba. And here's an amoeba. These are the protozoas. These are fungal spores. Yeah. The brown ones are fungal spores. I'm just wishing that I could have got a lot clearer. I'm going to take you out to 200, sorry, to 100. Take down the light. And I'm going to see if I can find you some nematodes, because those are also really exciting to see when we're looking in a compost to see what kind of nematodes might be in here. And we look at their functioning groups. So there's a whole bunch of stuff, as you can see. Lots of material, lots of bits and pieces. And when we learn to look, the things that we're looking for are the life in here. And these things that we're seeing, you know, again, here's another amoeba. It also looks like it's got little spikes on it. It looks like it's got spikes here and here. I haven't seen one of those before. But these are really important because they are part of the system. And above it there is also a little piece of fungi. This here is not a fungi. This here is a piece of the plant material that was in there that I know. But actually this might be, let's go and see closer. Let me have a look at it closer. It might be a microarthropod. Let me bring it into the middle. Because it does look like it might have a leg on it, in which case it wouldn't be in the middle. Let's take it right into the middle. I'll give you more light. Let's have a look. What is that guy? It's got a lot of spikes on it. It's a very funny thing. I've not seen that before. It's very spiky. What is that? I don't know what that is. It could be part of a microarthropod. I don't think it's an amoeba. I think it might be part of an insect's body. I'm now going to go into, this is now going into a much wider frame. Get more focus there. And the reason I'm doing this is so that we can actually quickly look at a much wider area to look for the nematodes, because they move so quickly. And normally when we're doing an assessment on a compost or on a compost or soil, the first thing we do is we do what I'm doing now, which is we scout the entire plate looking for nematodes. Now, and when you see them, sometimes they're moving extremely quickly. And then when we do find them, what we try to do is figure out whether they are good nematodes or bad ones. I know it looks like we're just looking at a very strange terrain for some of you that are seeing this for the first time. We'll just go a little bit slowly here. There's all sorts of things, all sorts of material. Sometimes if you leave your sample sitting for long, the nematodes tend to go down to the bottom. So when you're doing an assessment, you try to do it as quickly as possible after you've shaken the microbes off the materials so that you can see them. But I'm hoping that you'll find just at least one. I saw some fairly large ones before while I was looking through here. I saw some omnivorous nematodes, and also a predatory nematode, which is good. I didn't see any root feeders. If we're looking through a compost like this and we come across root feeders, then there's something wrong with our compost. You do not want to get compost onto your system that's taking root feeders are considered to be the bad guys of the nematodes. This here, that we can see in the middle, we're going to zoom in on it just so you can see. We've all heard about the world of microplastics and how terrible it is. That there is a bit of microplastic. Yeah? And that's just at 100 X. So it's terrible that some of that stuff just gets into, there's another bit there, gets into our systems. You can see it actually has a blue tinge to it. When microplastics get into our soils, microplastics get into our systems, and although many microbes can break them down, it's still not very good because of the chemicals and things that are in those plastics, as we know. The forever chemicals and things that are now beginning to damage people's health. And when these things end up getting carried through into animals' bodies, and we're not sure yet what those pathways are with plants, but we're beginning to see microplastics in animals as well, and the products that we're eating, and also in humans are saying that we've got microplastics in our brains, some of us. It's quite scary. Some of it's just from consuming microplastics and broken down in bottles. I'm going to look for another slide while I put the next part of, because I haven't found nematodes, but I want you to see nematodes today. So, but coming back to back to the medium that we're looking at, we're trying to make a very microbially rich and alive compost. I'm going to make a new sample with this, and I'm sure I'm going to find nematodes. But while I'm doing that, I'm going to take you to the first part of how to make compost. So there's two small videos I'm going to share today. The first one is talking about your kit, your safety, how you're going to put things together. And the second one gets into the actual making of. So let me get that ready for you now. This is coming from our proprietary masterclass compost for this class. And here we go. Let's share that. To make compost and what you need to be wearing to be comfortable. Throughout these videos, you're going to see me in my most comfortable attire. I love to wear very loose, very baggy, very pocketed dungarees because they stay airy. Composting is hot business. I always wear a pair of gumboots, sometimes shorter one, sometimes longer one, depending on how wet or how rainy it might be outside. And in my many pockets, I carry some of the other essentials. A hat. If it's really hot and sunny, you're going to need a hat. If it's really hot, don't forget some sunscreen. We don't want to be producing good food and getting cancer at the same time. That's why it's important to have all these pockets. Something to drink. There's nothing worse than a hard day's slog in the field doing something and you get thirsty and you don't have something to drink. So make sure you've got a liter or two maybe by your side. Composting is hot. It's hot work. Be ready for that. And finally, gloves. I always have gloves. Okay? So throughout these next few videos, you're going to see me in different versions of the same attire. I'll always have my gloves. I'll always be in the airiest clothes that I possibly have, the lightest, the loosest, the airiest. Now we're going to move on to the equipment that we need. It's a really good idea to have all of your materials ready before you start making your compost. So I'm going to show you these items one by one. Please do take notes, write them down. You can improvise, but I found this to be extremely easy. To begin with, a good pair of gloves, a little fork, a little spade, some wire, some clippers to clip that wire and to cut the other materials we'll be using, a tape measure for measuring out the fabrics that we need, a 20-litre bucket, just one will do, a big spade, a pitchfork, very good for turning, a watering can with a good nozzle, and a thermometer. Make sure your thermometer is a composting thermometer that will go up to about 100 degrees centigrade, and that is about 2 feet long. You're also going to need some poly mesh. This is chicken poly mesh. This will be what we will make the cage for the compost out of. A 3.1 meter long piece is good enough to start with, but you'll need to perhaps get one or two of those. Some plastic sheeting to cover the top of your compost to stop the rain getting in, and some shade net. The shade net you'll be using for the bottom of your compost on the little elevated platform that you'll have it on. You'll need two pieces of about 1.5 meters by 1.5 meters, and then a piece of about 6 meters long that you'll fold in half to wrap around your compost pile to keep the moisture in. Then the big things, the wheelbarrow. Wheelbarrow is always handy. And I use a plastic pallet for the bottom of my compost. It allows air circulation, it doesn't rot. You'll need to have two of those because when you've made your compost on one and you're changing and turning it and putting it into another, you need to have another one on standby. That's why you also need the two pieces shade net. And finally, I use this very big trough for mixing. You can mix by mixing on the ground on a piece of plastic, but you'll find that your pitchfork will start to make holes in the plastic, and eventually you'll have to keep replacing it, which is not very good. So this it's a big plastic trough is perfect. I can drag it around. It's very light to move. I can drag it around. Put my compost materials in there, mix them up, and chuck them into the cage. I'm now going to show you how to make the compost cage. Remember, we want the compost to breathe. It's an aerobic process. Although thermophilic, it will get very hot. We need the air to come in to breathe through the entire system. If it doesn't, the microbes, as they multiply and generate that heat, they use up the oxygen in the pile. Where there's no oxygen, it can very quickly become anaerobic. So we need the pile to breathe. The materials we're using, the reason we're using the polymesh for the sides, we're using shade net on the bottom of a pallet that allows air to come in and through, hot air risers. The sides of the polymesh allow the compost to breathe on the sides, and then we're going to cover it in shade net just to keep some of the moisture in. And eventually, the only thing that we'll put that is not breathable will be a piece of plastic over the top, held in with a rubber band to stop any rain or any other materials falling on the inside of the compost pile. Biosafety is extremely important when making compost. You're dealing with a lot of bacteria, animal manures, etc., etc. So you want to be safe. This is about the only time that you won't see me in gloves, is when I'm making the actual cage. I secure the shape of the cage using wire, different types of wire. You can use just simple electric wire. You can get a pair of snippers to cut the wire. I have it in about 4-inch long pieces that I loop into the polymesh and then twist around to form a little lock. I do reuse the wire over and over and time again, which is why I like it to be a wire that has got some sort of a rubberized coating on it. It's safer on my fingers. I don't like to pierce my fingers on sharp bits of wire, especially working around these environments of high bacteria. I make sure that the wire that I'm using does not leave little cuts in my fingers. And also, when you're working around the compost pile, you don't want it to scratch any of your body that is exposed, for example, your arms or your hands. I'm going to share with you a Sue hack. Once you've made your compost pile and you put your plastic on top, it can blow away. It can get rained on, and the rain sits in and it creates a pool in the middle that either collapses the entire pile or tips your pile over on the side or whatever. So what I've made here is a big band that goes around. I've used a piece of string that has just come off, recycled piece of string, reloved, reused, repurposed. Piece of string that has come off a hay bale, and a piece of rubber from a vehicle tire or anything else. So that acts now as a big band that can eventually go around the compost and hold that plastic down. You'll thank me later. Here's another hack. Get all of your materials ready beforehand, right? So with your wire, get your wire measured out. I basically take the wire, loop it around, get the measurements that I want. Get all these bits ready so that by the time I'm ready to wire up the cage, I have my pieces ready. So I do so, I clip it like that, and then I'll go through here very quickly, and through here very quickly, and I have now got a bunch of pieces of little bits of wire. So you might be tempted to use tie wraps. Okay, tie wraps are cool, they're easy to use, they're cheap to buy, but they're not reusable. So remember, we're talking about plastics. So I'm going to go ahead and build it and take notes. Roll out the mesh to get exactly 12 feet. It's a little bit longer than we need, but it helps to give the structure some integrity. And the easiest way to do it is to roll it out on the floor. Really easy and nice and easy to cut this polymesh. Much nicer on the hands than wire. There we go. Now we have our poly mesh. There'll be about 3 feet overlap, so you're getting a piece of net that is about 1 meter diameter by the time you start tying it together. And try to make sure that when you're securing the poly mesh, it is an exact fit. You don't end up with the piece being bigger at the bottom than it is at the top. The best way to do that is to secure one piece, then follow the line and secure the bottom at that side, and then you'll know that it's going to be flush all the way around as you secure the rest. These are actually a bit big, but not to worry. You can actually now do this lying down because we're following the exact lines. So what I'm trying to do here is to line up the squares so that the mesh ends up exactly flush down the middle. There we are, perfect. Flushed all the way down to the bottom. The ties on the mesh are on the outside of the cage because you want to access them all the time. So these ties are on the outside. Okay, and now we're going to secure this other bit here. So you can see that part of the cage is double lined. That just helps to give it a lot more structure. Integrity, sorry. If your mesh is too short, you might find that it's difficult to stand and withstand the weight of the actual compost. Remember, you're putting a lot of materials in here, about 1 ton. Thank you, Lynette. So here we are. What do we have now? We've got a loop, a really cool loop of net mesh that just about reaches the width of the pallet, and the pallet is 1 meter wide. You don't want it to go over the edge or drop materials out, and you don't want it to be too small. So this is about 1 meter, and let's have the measurement behind it. This should be about 1 meter in width. Yep, exactly. Well, it's 3 feet and a little bit. Good enough. So now we put the shade net on the floor to stop materials going down. Okay. Sometimes, and if your pallet is small, let the shade net go down, or let it be a little bit longer, so that when you're moving materials across into, when you're turning your compost, you can pull out that shade net to catch any bits that fall down. So you can have longer bits that you might fold into the edge until you start turning it. Next step, very important, when you're putting your cage onto the net, make sure that it doesn't move the net and end up with holes at the bottom. The next step that I'm going to show you is something you're not going to do at this point when you make your compost, but I feel like it's important to show you so that you understand. It's going to be wrapping the shade net around the cage, which you'll only do once your compost is in there. There's a chance that while I do this, the cage collapses because it just does not have the structural integrity perhaps to hold the shade net, the plastic on top, and a rubber loop around it. But I'm going to try and demonstrate it to you anyway so that you get the gist of what you need the materials for. Let's go. So here's the shade net. We take it around the edge, all the way around the frame. Make sure that it comes all the way down to the bottom of the cage. The width of the shade net is slightly longer than the cage, doesn't matter, let it just hang into the top for now. And then what we're going to do is just fold it back a little bit. There you go, that's normally what will happen only once the compost is in. Okay? Now, the plastic will go over the top to stop anything entering your compost. You could be under trees like I am here. You don't want new green material coming in at any point in time. Once you've made the compost, that's it. So the plastic cover also to stop rain. And then we've got this thing here, which is going to be fun to see if we can actually hold this up. It might just collapse, but just a little bit of fun, hey? Yes. Go, stand back. Whoa. There you go. So no matter what, no matter how much wind there is, no matter how much drama there is, your compost will always be protected and it will always breathe from the sides and from underneath. When you do have rain, what we do is we stick a stick in the middle of the compost, and that gives the entire thing a tented shape so that the water will still run off. I'm sure you're all really excited now, because now you know how to make the cage, you know how to make the structure, you know what the materials, the tools, etc. are for, and you're excited and gunning to get on with making compost now. So let's do it. Okay, and whilst you're watching that, I hope you all know how to make your compost cage now. You know about your equipment. Information will be in Canvas. If there are any questions or anything, please do put them in the chat. I have been chasing nematodes for you. This is the first time. I'm having a really hard time looking for these little buggers. But I found one, so I want to show it to you before I lose it. Is there another option for pallets? Well, you want your compost to be raised so that it can breathe underneath. Remember, hot air rises, so you want air to be able to go in, and you need it to be able to breathe from underneath. So some people use coarse material at the bottom. So it could be maize stover, maize cobs, quite a thick layer of maize cobs underneath. But the problem is when you're turning your compost, you'll end up with that stuff being a bit messy. So I like to say, try to some people use logs. So they put 2 logs down the side and then branches about this thick, and then some across the top, leaning together so they act like a floor. The air can still go through, and once you've turned the compost, you can easily make another floor as well. It does make a big difference than having your compost stand on the ground. It can be on the ground with worst case scenarios. No, hay can't work. Hay is part of the feed that you'll be putting into your compost. It's microbe food, and when microbes will eat it, you'll hear about that later. Now, let me quickly take you to the nematode that I have got for you to see. Where did it go? Let's see. Where did it go? Find it. Sorry. Very quickly. It's beautiful material on the wall. There it is. There it is. It's actually exactly where it was where I left it. So let me. There we go. Let me get into the middle there so you can see if it's moving. I'm trying to focus on it. Oops, it's gone out. It's gone up. And there it is. Here's the side of the glass. It's moving across. I'm taking it over there and trying to get it in focus for you a little bit. Really hard to get them in focus when they're moving so fast. But let me actually see if I can take it into. Here you can see. Sorry guys, it's moving away. Let's catch it again. There you go. You'll see some of its body. It looks very snake-like. Here we get a picture of its head a bit better. And this is what we do. We chase them around and try and look at their head and look at their mouth, their feeding parts. So that gives us an indication of what they are up to. Whether they're root feeders or others. Anyway, yeah, that's our nematode. It moves very quickly. We've gone back to 100x because at 400x we just can't see it. It moves, it's moving quite quickly. We're going to chase it around the slide. There it's gone into the corner there. Very beautiful little creatures. We want them. We want them in our soil. So we're going to stop sharing that now. Was that wow? It's hard to get them. Tomorrow I will, or not tomorrow, in our next class, we'll also look and see if we can chase down some of the protozoas. There's one other slide that I'm going to make for you before the end, but I'm going to take you to the second video on compost making just now. And the slide that I'm going to show you is a very cool one of pond water, some pond water that I collected today. I know, it's so wow. The creatures, these micro worlds, it's mind-blowing. So hang in here for the next video on compost making. You do have them in your homework. You can always go back to them. You can ask some questions now, but I want to get that other slide on of the pond stuff as well. It's mind-blowing. And some of these, of course, also end up in soil. So let's go to part 2 of the composting masterclass. So what exactly is thermophilic compost? Our thermophilic compost method entails deliberately decomposing dead organic matter in the presence of oxygen, so aerobe conditions, and reaching hot temperature thresholds of over 55 degrees centigrade. Good quality thermophilic composting results in a high presence of diverse soil microbes and metabolites that we regenerative farmers use to inoculate and regenerate our soils and revitalize our crops. You can use your compost in different ways, as a solid compost, as an extract, or as a tea. Remember, it is an inoculation of soil microbes, so your fungi, your protozoa, your nematodes, your bacteria. If you don't have good levels, if you don't have good balance in your compost, your original compost, you won't get the good balance in your teas or your extracts. So it's fundamentally important to get your compost, solid compost, made extremely well and with very good and balanced microbial diversity. Composting is like cooking. You're going to need a good working environment, a good recipe, quality ingredients, great execution of that recipe, the right temperatures and moisture, of course, mixing when required, and then, of course, settling before consumption. So you're going to want to make your compost in a cool place that is protected from wind, from rain, from too much sun. You want your compost piles to be made close to where there's water and also close to where you're going to be using them so that you don't have to be trudging your ready-made compost over long distances. So we're making our compost to grow microbes, in particular our bacteria, our fungi, our protozoa and our nematodes. When we look at why we're doing it, it could be because of our next desired crop based on the succession and the fungal to bacterial ratio, but it could be also because our plants are suffering from pests and diseases, which indicate that our soils are missing a good solid soil food web balance. And speaking of balances, in order to get the bacteria, the fungi, the nematodes and the protozoa into our compost, we need to make sure that we're giving them the right food. It's their party, right? So we start off with a party food, which is to encourage the bacterial activity. The bacteria come in and chomp on that very high nitrogen bacterial food and in the process multiply and drive up temperatures very steeply. In the process also they begin to consume a lot of oxygen, and that's why when we reach some of those temperatures we need to turn the pile to bring oxygen back in, but also to change the sides and the other materials and bring them into the mix. It's the core of the compost that will actually have that be like the furnace of the compost. The green materials are burned slower, so they keep the temperatures right up in the thermophilic phase that we want. We want 3 turns at above 55 degrees, and we'll talk about those thresholds later. Minimum of 3 turns throughout the duration of making the compost, and as the compost cools down, the other microorganisms will come in to predate on the bacteria, and we'll see the maize cob and cow manure. Your cow manure has much more nitrogen in it than a maize cob. So when we look at the list of our party foods, manures are high in those lists, and all of the manures have different relative amounts of nitrogen. It's advisable to work with the manures that are closest to you. It's going to be much more cost effective, and also you're going to have biology that is native to your area. You can see that horse manure has less nitrogen than, say, chicken manure. And you also find that you might have some high nitrogen ingredients around you that are not on the list. For example, I use black soldier fly frass, which has got a very high carbon to nitrogen ratio. What I advise you to do is have a look on the internet, and you'll be able to look at those carbon to nitrogen ratios. And typically you're trying to look at things that have a carbon to nitrogen ratio of less than 10. Less than 15, maybe 15 to 1, 10 to 1, something like that will give you some good party food. For your greens, you're looking for a carbon to nitrogen ratio of on average around 30 to 1. It could be higher, right up to even 500 to 1. But what you're looking for essentially are materials that have got greens in them. You can also use in your greens compost that has failed, so spent compost piles, and a bunch of other things, including your kitchen waste from your kitchen, your green waste from your kitchen. But typically your greens can be your weeds, your green grasses, your garden waste, your vegetable waste, fresh stover, maize stover. As long as it's looking green, is lush and a little bit green, you can count those, consider those as your greens. So green grass that has dried as green but has now gone a strawy-ish color but with tinges of green in it is still considered green. The nitrogen was still in it. It was still a more of a green input than if it had dried, for example, like straw dried in the field and gone through senescence. So think about that when you're thinking about your grasses. When you harvest them, you don't want to put them in a big pile because they heat up just so very quickly, but you can lay them out and let your grasses dry as green and still store them then dried and use them as green. Your browns generally are very high in carbon, so those are either plants that have dried, so maize stover that may have dried would be considered a brown, wood chips, twigs, cardboard, paper, straw, all of those materials that have gone through their growth period or are just very high in lignin and in carbon. There are a few considerations you want to take about the ingredients that you're using, so let's look at party food first. What is a carbon to nitrogen ratio of the party food that you're thinking of using? If you don't know, have a good look on the internet, there's plenty of information there. What was the animal's diet? An animal that is fed on grain will have a higher carbon to nitrogen ratio, more nitrogen in the end product than an animal that is not fed on grains. It will be more party foodish. How fresh is the manure? Old manure is less party food than fresh manure. Your bacteria won't have such a big party in old manure. What's the likelihood of dewormers? So dewormers will affect the nematodes, and you want nematodes to grow. They're part of the soil food web. What is the likelihood of antibiotics? That will also affect the organisms, because antibiotics actually are there to kill bacteria and to kill microbes. What is the likelihood of any other contaminants, sprays, etc.? And then finally, considering the denseness of the weight of your party food. An example is something like horse manure versus black soldier fly frass. A bucket of horse manure and a bucket of black soldier fly frass, and might both be in terms of volume the same. The black soldier fly frass will be much denser, much heavier, and when I use it sparingly, so I use 5% of black soldier fly frass and then increase the carbon by an extra 5%. So you don't want your compost pile to fire up and keep burning away over a long period of time, because you'll have to keep breaking it down to stop it becoming anaerobic, running out of oxygen, and it'll be a lot of labor and a lot of work. So let's look at our considerations for greens. We want to know how old is the green material, how long has it stayed, how long since it was harvested. A very young, say, clippings of a lawn versus long grass that has already seeded versus grass that has still got the seed in the stalk. Each of those will have different reactions in your compost pile, so you can practice with the different materials you have and see how they contribute to the long burn. It'll be based on those differentials. Could there be contaminants in the materials, pesticides, etc.? How quickly will they heat up if they're left in a pile? So sometimes you can use that as an indicator. If you want to know if your grass is going to be good in keeping that slow burn, put it in a pile and see what kind of energy it has in it in terms of its heating capacity. Sometimes you can put your grass in a pile and within a few hours the temperatures are really high. I had mine going up to 65 degrees just within a day. Are they cut up enough, or will they take a long time to break down? Yeah, so make sure that your materials are cut down so that the microbes can get at them easier. At the same time, you don't want the grass to be clumping up, and that's why I say when you're mixing your grass in your pile, mix it in as a dry material so you can fluff it around and it isn't all clumping together. When you get your grasses and things or leaves clumping together, they can very quickly turn a pile or create anaerobic pockets in a pile because the oxygen can't get through. The microbes that will survive in those conditions will be the anaerobes, and we don't really want them. So finally, looking at the brown materials and the conditions we need to look out for. How dry are they? How much soaking is needed? Remember, your compost pile is going to take a few months to break down. If you don't have your materials saturated enough with enough water in them, at the 50% which we'll talk about, then they will dry in the middle and the microbes won't grow. So how dry are your browns to begin with, because that will influence how much soaking is needing. Could there be contaminants in the materials, pesticides, etc.? Are you using wood that has been treated? Not good. Are they the right size to encourage porosity? So your browns actually bring in two aspects, not only fungal food, but also aeration, because you don't want to have finely ground sawdust or wood shavings. You want to have wood chips so that as they're moved around, as they're turned around, they also create a fluffier and more breathable if that's the right word to use, compost. And then you want to know that they're not too dense also, as I've just said, that they could cause anything like anaerobic pockets, as I've explained also with the clumping of grasses. Super, so we're finally at the recipe stage. We're going to do a 47 by 20 litre recipe. That'll give us about 940 cubic litres. The 10% will be 4.7 buckets of party food, 14.1 buckets of green and 28.2 buckets of browns. Get your materials weighed out, get them ready, put them on the side before you go on to the next steps. The day before you make your compost, make sure that you make your cage so that you're not faffing and trying to get that done all at the same time. It's a lot of work, as I've said before. If you don't know how to make the cage, just go back to our lesson before this, the introductory to this composting masterclass. The evening before you make your compost, soak all your brown materials. So take all your wood chips, all your cardboard, or whatever the materials are that you're using that are brown, and soak them. I put mine into a bath of water, completely drown them. I do the same thing with my straw. I put the straw into water, step on it, mush it down and leave it overnight from 6 p.m. Until early morning, 8, 9 o'clock in the morning. I chuck away the water from the straw, by the way, because I don't know where it was produced and it could have inherent pesticides that I don't want to bring into my pile. So I also use this as a method of rinsing out any potential pesticides in the straw. On the day of making your compost, you've got 3 things to do. Try and do them in this order. Get all your dry materials out of the water and let them drain. So just put them on the grass on the side, or I put them on a plastic sheeting and let them drain. I then start getting my greens ready, either raking those up that I've cut and had naturally dried out in the field rather than in piles, or I start mowing the grass, or chopping up if I'm using maize stalks or anything, chopping them up then into smaller bits and pieces. And then take your party food and pour some water in it and bring it up to about a 50% wet, sort of like slurry. Now divide all of the materials into 4. You're going to be mixing in batches of 4. This will be less labor intensive than trying to mix the whole lot in one go, but it will also help you to get more uniformity across the entire compost pile. You're going to have 1.2 buckets of party food, 3.5 buckets of greens, and 7 buckets of browns for each of your batches. Put them all into your trough or all into the area that you're going to mix them. So now is a real critical time. You need to bring your batch 1 up to a moisture level of about 50%. So to measure that 50%, you'll be adding in water incrementally, slowly as you mix and turn and get all of those ingredients mixed into a lovely homogeneous mix. You'll take a handful of the compost out and squeeze it. At 50%, you'll see a few drops appear just between your fingertips. If they drop and you get a lot of drops falling from your hand, that is well above 50%. And if there's no liquid at all that seeps through your fingers there into that one drop, your compost is a little bit too dry. Keep adding water incrementally until you get it as close to that 50% as you possibly can. Take your first batch and fill it into the cage that you've got ready, and continue the process with the next 3 batches until you've filled it up to the top. Composting is really hard work, so get some help to make sure that you keep that consistency throughout the whole process. Once your compost cage is full to the top, wrap the cage with some shade net to keep the moisture in, keep the wind out. Put a thermometer in and take your first reading, and put a peg with an ID in it, so that in case you're making more than one compost, you don't mistake which is which. Then take some plastic or some tarpaulin, put it across the top to stop anything getting into the compost pile, anything falling from above, or any rain. At this point as well, you'll also want to put a band around to hold the plastic down so that it doesn't get blown off. And you might want to put, you'll pull up your thermometer. It will act as tempt the plastic a little bit, so that if it does rain, the rain will just run off. And that's it. Next up, record keeping. So that is the heavy lifting around composting, knowing your materials. Edith, you said the sound had gone. Had the sound gone for everybody or just for you?
No, the sound was good all through. I think somebody experienced bad internet there.
Okay, there's also some parts of the video where I'm not speaking. So, and I'm not voicing over, you can just see. Are there any questions about your recipe? Any questions at all? Please do put your hands up and let's have a discussion about that because it's so important we get it right. You can go back and look at it in your homework, but or maybe you can just show me with thumbs up if you all think you've got it about the recipes, about having the right percentages of carbon, browns, greens, and party food. Let's see a show of hands. Has everybody kind of like got that right? How to make the cage and the idea around the party food. Edith, you put your hand up to ask a question, please. Let's give Edith the microphone. Go ahead.
Hi, this thing about the green grass, we're told that normally even cows, you don't give them grass that you've cut with lawnmower. Does it affect the compost? Also, would it affect a lawnmower? It won't affect.
No. I cut my grass with a grass cutter.
Yeah.
Lawnmower, and I use that for my compost. What you want to do it when your grass is not seeded, because by that time the grass has got old. It's got most energy in it before it goes to seed. That stage is what you want for your compost because that allows it to continue burning for a long period of time. You want the heat in your compost ideally is going to be operating. The first threshold will be between day 3 and 6 and last for another maybe 3 days. So you're talking about 9 days. The next 2 thresholds are equally going to be about 3 and 3 days. So you're going to have your compost heating up for about 2 weeks. The energy in there needs to be enough to keep that heat going. So you want to make sure that the quality of ingredients is not just a whole bunch of spent material that's got no energy in it. Yeah.
How about the types of grasses? For example, what we call Kikuyu grass vis-a-vis this silky grass that is so even doesn't cut with a lawnmower.
Any grass that is green.
It's called watergrass. The watergrass, I think.
Oh, you're talking about the sedges.
Like the
sedges. So the sedges that are normally around water areas, that you're right, they don't cut very well because they're very fibrous. They are also very waxy.
Yes.
Yeah? So they are not going to be too bad in small volumes, but they won't be very good in big volumes because it takes more microbes to break them down because they are already quite fibrousy. They don't break down very quickly and they're very waxy. So the microbes have to be there in either larger numbers or they have to work harder. Yeah.
Okay. And the haze, rice haze vis-a-vis the other grass haze.
So let's look at what hay is compared to straw. Okay. Normally, when rice grows, the rice seeds itself, those are the fruits. The plant is already spent by the time those are harvested, right?
Yeah.
So that is straw.
Okay.
It's normally a lot of lignin in it. People will try to sell it as hay for people to feed their cows, but it's just fiber.
Yeah.
That would be used as a brown, high carbon. Yeah. Straw itself, high carbon. If you're going to use a grass that was a tall grass just before it's seeded, the energy is still in it, that could be used for your green.
Oh, okay.
Sometimes,
Okay.
sometimes it's about the age of the plant, and sometimes it's about the color. So if we look at green leaves on trees, if they're old trees with old green leaves, those leaves are much more carbony and high carbon than they are young leaves that are green and more nitrogen. Yeah.
Okay. Okay. Thank you.
And with grass that is green, that is young, so hay for cattle should be cut when the flowers and the seeds are inside, because that's where the energy is for your cattle.
Yeah.
Then when it's dried, so it might be looking a little bit green, but it's not that the seeds have come out and they've flowered, that it's straw. So the best hay for cattle will have some tinges of green in it, and if it's hay from something like lucerne, it's even greener. But the reality is a lot of what we feed our animals and we're sold for our animals is not hay, it's straw. It is grass, straw. It is old. It is seeded. Somebody's collected the seed, or the seed has been dispersed. The flowers have already come up. They've seeded. And for people that have cattle, it's very important to know the difference, because it's the difference between feeding your animals just fiber and they don't get enough energy for their body needs, or for their milk production, or their raising of their calf if they're pregnant, versus just eating fiber. Yeah.
My cows are very proud. They don't eat straw. They must eat hay.
Perfect. 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 They know. If they had a difference, they would know, but they don't have the choice. And many people, especially as it gets drier, end up selling straw. Thank you.
All right. Thank you.
So I can see Joyce has said, I use wooden pallets as a base. Is that okay? Yes, you can. And you will find over time they will begin to get eaten by the microbes, because remember, they're also wood. So the fungi will try and eat them. So in between using them and moving one pile to another, make sure you dry them in the hot sun so that the fungi then can't exist, because the fungi does need moisture. Otherwise, they'll just start to chomp on that as well. Evelyn, you said, can chicken manure be used in compost making? Absolutely, yes. Chicken manure is quite high in nitrogen. It can be used as a party food. The question is, what else is in the chicken manure? So a lot of people will collect their chicken bedding with chicken manure and say this is chicken manure. It isn't. It is chicken manure and bedding. And if that bedding is high carbon, if it is wood shavings, if it is straw, if it is sawdust, whatever it could be, there is a high percentage of carbon in there that is not chicken manure. So if you want to know, if you've got chicken manure that is mixed chicken litter, sieve it. Get a coffee mesh, that's the small mesh wire in a frame, and sieve 1 bucket and see what percentage of pure chicken stays behind and what percentage is actually that wooden stuff. Or the other way, maybe the chicken stuff will go through and the woody stuff will stay behind. And then you'll know in your mix what percentage is actually chicken manure and work with the right percentage of chicken manure, okay? And the woody stuff that's in the bedding, you can allocate to your carbon, to your high carbon. On Tuesday next week, we'll be looking at the record keeping around this and how to put in those correct records. But start thinking about that now. That when you're looking at manure, you're thinking about that stuff that came out of the animal without it being contaminated or mixed with other stuff. Selma, the same thing about your rabbits, okay? All the manures from animals will be very good for party food. Joyce, your hand's up, go ahead. Let's give Joyce microphone. James, have we managed to unmute Joyce?
Yes, please, Joyce, kindly take the request on the screen to unmute. I've given you.
Okay, thank you so much. I have one question. On the party, there were several of them. So are we going all of them, or just one or two, or a minimum of 4?
You want to use whatever you have that is available. Okay. The regenerative agriculture is not a form of agriculture that is switch out chemical agriculture for a more natural way. It's also a way of looking at what are the materials around you. And the reason is within your environment, outside, in your ground, on your fields, in your farm, you've got your own microbiomes on your plants, in your soil, etc., that are used to your specific farm that are very different across other people's farms. If you've got livestock that are grazing and being reared in that environment, the chances are that their manure is going to be better to use than in-bought manure. You can, if you want, if you don't have the right volumes, do a mixture. What you're going to find, as I said earlier, is many of your first composts will fail. And when they fail, talk to us about it, because we'll be saying, where was the failure point and what drove it? Sometimes it's because there's not enough party food. It's not strong enough. People are using manure that has been sitting outside. It has been rained on. It has been leached. It doesn't have the energy in it or the nitrogen in it that it needs. Sometimes it's because they didn't use the right volume, or they had a lot of bedding so that the carbon component, the browns component, was too high. It wasn't 10% of the manures. I use, like I say, black soldier fly frass all the time now because it is very foolproof. It's very conk, it's very good, and it gets my temperatures up quite high quite quickly. So yeah. So the turning of the compost, it's going to be done according to the thresholds. We only want to turn about 3 times. Okay? And those 3 times are when the temperatures are right and when the temperatures have sustained for long enough to kill any seeds or any pathogens in the mix. Next Tuesday, when we're having the class around record keeping, we're going to talk about that, because there's a lot of documenting, and as you document, you'll begin to know where those turning phases are. And once you follow that repetitively for a while, you'll soon be able to, and if you're teaching somebody, you'll soon be able to get it right with your eyes closed almost. When you've got a good grasp of how your materials react together, how they react with the ambient temperatures, if it's very cold outside, if it's very hot outside. If it's very, very cold outside, you'll find that you'll actually probably need to use more party food. So if you're going into, for example, our winters are June, July, you might find that 10% party food, the temperatures were good, but you didn't make 3 temperature thresholds after the turns, and therefore you need to increase that up to maybe 15%. We'll get to there Tuesday. Don't worry, you do not need to make your compost this week. You do need to go back to those 2 videos and start getting your things ready, really understanding it. And on Tuesday, we will pick up and start talking about the next steps. Judith, you said, what if I have bio-slurry? Can I use it as an ingredient, and how? Your bio-slurry is will be very good actually in terms of feeding the microbes and bringing the temperatures up, even though it is a lot of microbes itself. I would use my bio-slurry to do the soaking of my ingredients that need soaking, and I would be using it as part of the water mix. So instead of adding just plain water, I would rather add my bio-slurry in there. Remember that bio-slurry has come from a very anaerobic environment. So the microbes in there are anaerobes, and what we're trying to do is create a lot of aerobic microbes and create it for an aerobic environment. When you put bio-slurry in a compost that you're making aerobic, the microbes will die. What they will release as they die are all the nutrients and the minerals that they've had in their body, and they will now become available for the for the aerobic microbes to consume and build their bodies with and multiply with as well. Okay. Now, does anybody want to, is bio-slurry do the same role as compost? No, very different things. As I've said, bio-slurry is an anaerobic process. Bio-slurry ends up with a lot of dissolved nutrients, which might make plants grow very, very quickly. But in terms of bringing the aerobic microbes into the ground, not really. Yeah, it will bring minerals and dissolved minerals, and bioslurry can also, depending on what went into the bioslurry, some people put a lot of other materials, not just livestock waste. And sometimes those materials also bring in, depending on where their source was, if it's market waste, it could bring in some of the other toxins that have come in on the products that were grown. They could be pesticides, etc. So they might, you need to know, you need to think back to what is it that you're putting in and how might it affect very fragile microbial life. So if you brought in even when like I said, when I'm bringing in straw and things like that and I'm soaking them, I'm not adding that water back in the compost. There's no doubt there would be glyphosate and other chemicals and maybe pesticides in that water. And if I bring that into my compost, it is likely going to affect the biology that I'm trying to grow. So bioslurry, depending on what it's come from, if it's just livestock manures, if your livestock manures and they've had a lot of antibiotics and worming medication, then yeah, might not be as good as you want it to be. How do we deal with microplastics? Well, you try not to get a source of material for your compost that has plastic in it. Okay. Sometimes if you've got things on your farms like bags or sisal string is okay, it biodegrades, but plastic twine, and that breaks up into little plastic bits. That's how these microplastics get into things. Generally speaking, the microplastics won't get into the actual bodies of the microbes. Some of them can break them down. They do have that ameliorating and a remediation aspect with plastics. But what you don't want is to be bringing garbage into your compost. The compost making is not recycling garbage. A lot of people think that it is. It isn't. It's actually making something specifically to develop your microbial community and then inoculate your soils and your plants. Does anybody want to go quickly onto the microscope to have a look at what this is? What settled in the bottom here is some algae that was in my pond floating outside in the water in a bucket that I have. So remember, you know, you leave your water outside and it goes a tinge of green. Have you ever wondered what that looks like? Do you want to have a quick look? You put your hands up if you want to have a quick look. I'm going to go straight into that. So for those that do want to have a quick look, just be on standby. It is so cool. And for those that don't, it was great having you in class today. And I hope to see you all on Tuesday when we continue with our discussion about compost. But right now, I want to show you this stuff is so super cool. So these little guys, you think that green stuff is just floating around is, I don't know, some sort of plant. And in a way, algae cross that barrier between being plant and animal. But when you see them under the microscope, these guys, they're called euglenias. There's euglenias and a few others, a bunch of other ones as well. I'm going to just quickly do that, switch over to the scope, and you'll see it's actually really, really cool. Put that on box here. And some of these are used and dried. We've all heard of spirulina and the likes. Some of them are used in food products. Some of them are used for, all sorts of things. But for us, right now, we're going to there we go. Let's just see here. Let me quickly. They are okay. So this is what they look like at 10x, at 40x. Sorry. Okay. See, some of them are like moving around. I'm going to now take us into, and give you more light, more focus. Some of them are moving and you can see them. This is what they look like at 100x. So let's go into 400x. Check these guys. It's quite deep, so there are some swimming on top, some moving around. These are Euglena. They are microalgae. They've got a little string on the sides of them. You can see that on their bodies. They are protozoa, but algae. So they've got that little string on them. You can see it's the flagellate and the flagella. They are flagellates, and they will use that to spin around and move around as they're doing. Some of them, you can see some of the flagella there in the bottom, down here in particular. Let's see, down here, these flagella right here are floating around without the bodies of the things. There's another one in here. But you can see some of them have them on them. You see some there. There's one over here that's connected to that guy. And those ones that are moving, that's how they actually move with that thing. They move in an erratic way, and you can tell that they are protozoa because of the way that they're moving. And that's based on the fact that they don't have, of the protozoa, they're not like ciliates. Ciliates have hair all over their body, and they are like, in fact, they're so hairy, and each of those cilia, they move with them, they move very quickly. But these bumble along because they are protozoa. So when you're looking next time into a green pond, sometimes it can be because it is filamentous algae, and sometimes it's because of these guys. And they will appear literally out of nowhere. They are the first photosynthesizers. These are the little guys 400 or whatever million years ago. They're the first ones to appear in a water body even before the filamentous stuff, and they start using the nutrient in there, and they start doing that nutrient cycling from them. Many of them are now being thought to be superfoods in concentrated and powder forms. You've heard of spirulina, chlorella, and even Euglena. Some of them very high in those micronutrients that you don't get. So literally, these are pretty cool, right? These are the first little creatures that appear in water. Even if you leave a glass of water outside, the first things you're going to see as it starts to go green are these tiny things. And on that note, we will say bye-bye. Edith, is your hand up again quickly? Just give me a thumbs up or not. James, meanwhile, you can be giving us all our microphones and our cameras. Edith, go ahead if you've got another question. And I hope you guys all like the micro world. I'm looking under my microscope all the time. I have seen the most amazing things. And more species of micro life than there are anything that you've ever seen. It's just incredible. James, are you there? And we all have our microphones and our cameras. Edith, please put your question into chat. Everything okay?
Yep. Very intense, but very good one.
Great.
Very intense.
Great.
I used to love biology before I ran away.
Wonderful.
Yeah.
Okay,
Thank you.
super cool. We'll see you all next week. So bye-bye and take care, everyone.
Thank you.
Bye-bye. James will give
Bye.
you information about the thermometer, Judith. Bye-bye.
Yeah.
Okay, I will.
Bye.
Bye.
Bye-bye.
Thank you.